Okay, welcome back to Lecture 4. I am smashing out all these lecture videos in like one day right now because I really don't want to be doing these tomorrow if I can help it because I've got some other homework to do. But we're going to do Lecture 4 right now, and hopefully we're going to finish it in around 30-40 minutes, hopefully less than that. But this is just about quizzes, stuff like that, gated transport, a couple other things to keep in mind. So yeah, I feel like these are things I've already talked about before. Already mentioned this before. How do they make choices? How does a protein make this choice of where to go, stuff like that, and it's all decided by signal sequences. And I already talked about this slide. These are the important sequences to keep in mind. This one, not so much. Don't think it's that important. This one, definitely important. This one, definitely important. This one, definitely important. And this one, definitely important. So, yeah. Oh, and here we go again. There you go. So, I'm going to be honest with you. Drank too much water. They usually don't ask you, like, experiment questions of, like, oh, how were the sequences discovered? So I'm not going to spend time on this, really. This is not that important to me. They mentioned it in lecture. I can't remember the exact reason or how that was discovered, but I do remember that it wasn't very useful when I took the class. I think the main things that I want you guys to take away from this lecture is the systematic approach to how this is done. So, yeah, I'm not going to bother on this too much. If you want to learn more about this, go back and watch the actual lecture. I'm sure Professor Khan explains it far better than I ever could. So please go back and look at the lecture if you want like an in-depth explanation about how this works. But I don't think this is something that is going to be tested. Like, how did they discover that signal sequences were a thing? Yeah. What I do want to talk about, though, is this specific sequence here, this hydrophobic one. This specific sequence is a sequence that tells the protein that it needs to be imported into the ER. And so that's where we're going to pick up with these. So this entire process is talking about protein translocation from the cytosol into the ER. That is the whole point of this section. We're specifically talking about how do we get proteins from the cytosol into the ER. So, protein translocation requires a, just smacked my mic, requires a signal recognition particle to direct the, ER signal sequence to a specific receptor. And so they show all of this stuff. What I want to make clear is basically what's going to happen, right, is you have your mRNA. Your mRNA is going to be read by your ribosome and you're going to produce a protein, right? At the very start of your protein, or somewhere in the protein, you're going to have a region that is just something like this. Doesn't have to be this. It just has to be a bunch of hydrophobic residues. And it needs to be near close to the N-terminus, generally speaking. When that hydrophobic region is printed out of the ribosome, because remember, the ribosome is reading the mRNA and at the same time is producing the protein, when that sequence comes out, that hydrophobic sequence comes out, what happens is the SRP, the signal recognition particle, which is basically a protein mRNA, it's a protein RNA thing, complex, like the ribosome is, that thing binds to the hydrophobic amino acid sequence and basically halts translation. And it halts translation until it carries the entire complex to an SRP receptor. So here you can see here, look at this. So if we look at that, see this here, this portion recognizes this portion, and it leads to this complex, right? So after this binds to here, it pulls the ribosome to the SRP receptor, which is located on the ER membrane. And so once it binds to the ER membrane, then we can focus on the next steps. But like I said before, the SRP binds to the ribosome and halts translation. Why might pausing translation be important to cell safety? Long story short, you don't want proteins that are supposed to be on the outside of your cell or to be secreted from the cell to be inside of your cell, right? Plain and simple. It basically just prevents error. It prevents the possibility of this protein accidentally getting stuck in the cytosol. It basically ensures that it's going to go to the ribosome. Sorry, it's not ribosome. It ensures it's going to go to the ER. It ensures it's going to go to the Golgi. It ensures it's going to get secreted, basically. It's basically a way of confirming that, this protein is not going to get translated in the cytosol and mess up other processes. So, this SRP is made of a couple things. There's a translational pause domain. There's a hinge, which is like it's like your arm. It's like the elbow of your arm. And then there is the signal sequence binding pocket. This section binds the hydrophobic region of the protein that comes out of the ribosome. This region binds to the ribosome and stops it from continuing translation. And this region is just to make sure that the protein's plus RNA complex thing is flexible. And this is kind of what the whole thing looks like in a 3D model. So anyways, once that's happened, so once this is bound and all that, you're going to scroll down here. So you can see here, we have our signal sequence coming out of our protein. We then bind our SRP to the protein, halting translation. Then we bind our entire complex to this SRP receptor. And when it binds, what ends up happening is this SRP plus the receptor plus other proteins feed this protein into a protein translocator. So basically this SRP receptor is right next to this protein translocator. So when the SRP meets its receptor, it basically binds the translocator immediately. And so after this ribosome is basically handed off to the translocator, the SRP receptor dissociates from the ribosome along with the SRP, and then these two split, and then this can be recycled for this process to occur again. Now, moving forward, once this ribosome has been attached to this translocator, we are at this point now. Basically, the hydrophobic sequence hugs one side of the translocator, while the ribosome continues to feed the protein into the ER lumen. And, remember, as soon as this SRP is removed, translation can continue. So this is removed now, translation can continue. This protein basically becomes incredibly long. And so eventually what happens is once translation is finished, this red sequence here is cleaved off and the protein is released into the ER lumen. And so this is just a cool little image showing how this can actually happen multiple times to the same mRNA. So it's just a really cool process, very multifaceted, can happen a bunch. Here is the structure of the protein translocator. I don't really know if there's much for me to really talk about here other than the fact there's the plug in there. But yeah. Here's a cool little thing about how this works. So basically, remember the ribosome, the start sequence, this transfer sequence here, it's probably halted, right? It's halted before this. This then binds and opens this translocator. And then once it's in there, after translation is finished, pretty much, this signal peptidase basically cleaves this section of the protein, and the rest of it is released into the ER. Now, what happens if there's multiple domains or hydrophobic regions in the protein? Well, remember, hydrophobic regions of the protein will interact very well with the phospholipid bilayer because the phospholipid bilayer is also hydrophobic. And so what's going to happen is the first hydrophobic sequence, as usual, just binds here, and we keep translating. We keep translating, and eventually we're going to get to the point where this hydrophobic region, this orange region, is also going to reach this kind of middle area where this translocator is. That translocator is actually going to recognize that. And what happens is that the signal peptidase cleaves right here, and basically this portion flops down, and the rest of it is just there. So now we have a case where it's a transmembrane protein. So you can see here, C-terminus is in the cytosol, but the N-terminus is in the lumen. So this is very important to keep in mind. If there are two hydrophobic regions like this, you can get a transmembrane This also works with anything above two transmembrane domains. So you can have 3 hydrophobic domains, 4, 5, 6, 7 if you want a GPCR. There's a bunch of different ways that you can do this. So this kind of thing here, where it's like a transmembrane protein, can also occur if the start transfer sequence is in the middle of the polypeptide. So you see here's an example of that. Notice how it's in the middle. There's no cleavage in this case. Basically what happens is the entire protein is translated, and so once the whole thing is translated, this whole thing plops through, and the entire C-terminus ends up on this side. Now, the one thing I want you to remember is the side of the protein that has the most plus charges, or the most amount of basic amino acids, so not histidines really, more so lysines and arginines, that will end up on the cytosolic side. So if the N-terminal side has more plus charges, the N-terminal side is in the cytosol. If the C-terminal side has more plus charges, then the C-terminus is in the cytosol. So notice how this and this are slightly different, right? Depends on which side has more plus charges. Is it the N-terminus or the C-terminus? So plus is in the cytosol. Remember that. Plus in the cytosol. That's one of the things you should always be looking for when you're looking for these proteins. So now let's talk about multiple pass transmembrane proteins. So here we have a start transfer sequence and a stop transfer sequence. So there's no cleavage in this case, right? Because it's not close enough to the edge. It needs to be closer to here for there to be cleavage. So here we have this first one goes in. We're going to translate. When we're translating, we're going to put this guy in here. And then we're going to keep translating, and we're going to hit this second hydrophobic region. We're going to keep translating, and we're done. That's it. There's no cleavage. So we just end up with this. That's it. So now we have to ask ourselves, where are the plus residues located? And I know what you're saying. The answer's already given to you. But always look at the first transmembrane pass to determine where the plus charges are. So the fact that the N-terminus is in the cytosol means that the plus charges are on this side. So they should be at the N-terminus. Lo and behold, that's the answer. Now, for multiple pass transmembrane proteins, you've got hydrophobicity plots. I think you guys have seen these before. Basically, if it's above if it's above the middle line, usually the middle line 0, it's hydrophobic. If it's below it, it's hydrophilic. So you can see here we have 1, 2, 3, 4, 5, 6, 7. So that means there's 7 transmembrane passes. So 1, 2, 3, 4, 5, 6, 7. This is what G proteins look like. G protein coupled receptors are 7 transmembrane pass proteins. And this is an example of what it would actually look like in like a protein, basically. So you can see here, we have a start sequence, we have another membrane pass section, another membrane pass section, another one, another one, another one, and another one. And we can tell what the orientation of this is because we can see that the plus is on the C-terminal side. So this C-terminal side should be in the cytosol, while this N-terminal side should be in the lumen. So this is the start of the protein we can see here. See how this is in the lumen and how this is in the cytosol? The C-terminal side is in the cytosol. That's very important to keep in mind. So now, let's take a look at this. Where are the N and C termini? Well, there you go. But that's not what the question's asking. How many transmembrane passes are there? Well, this is not super close to the edge, so it's not going to get cleaved off. So we're going to have 1, 2, 3, because these are all hydrophobic. Okay, so this is the first sequence. So this is going to decide the orientation of the protein. And let's look for the basic residues. So if we look through this, the only basic residues I see are actually here. And so what that tells me is that, okay, the basic residues are on the C-terminal side, not the N-terminal side of this hydrophobic section. So I expect the C-terminal side to be in the cytosol and the N-terminal side to be in the lumen while having 3 transmembrane passes. Is the signal sequence cleaved? Nope, because it's not close enough to the edge. It needs to be closer to the edge for it to be cleaved. Is the N-terminal side of the protein lumen or cytosol? We said that the N-terminal side is going to be more lumen because it's because these R's are more towards the C-terminal side. How many transmembrane passes are there? There's going to be 3. 1, 2, 3. Remember, there's no cleavage, so it's going to be 3 of them. And lo and behold, here we go. See, this is exactly what we talked about. 3 transmembrane passes, N-terminus in the ER lumen, C-terminus in the cytosol, right? It's exactly what we talked about. So now we have to think about this. If this protein is transported to the plasma membrane, is the N-terminus extracellular or intracellular? Well, remember, what's in the cytosol stays in the cytosol. So we can see here, the N-terminus is not in the cytosol, it's in the ER lumen, which means that it's going to end up on the extracellular side of the cell, not intracellular, once it's in the membrane of the cell. So, extracellular. There you go. See, because remember, what's in the cytosol stays in the cytosol. So this plus side of the protein would remain in the cytosol. Okay, so now let's look at this. What happens if the N-terminal end is shorter? How many transmembrane passes are there? Well, notice how this is very close to the edge. This is going to get cleaved off because it's super close. So this is going to get cleaved off. We're going to have 1, 2 transmembrane passes. There you go. Yeah, and so basically the way that you would keep this in mind actually is the N-terminus would be in the N-terminus would be in the lumen, if I'm not mistaken, and then the C-terminus would be in the lumen as well. Yeah. Yep. So if you look at this, right, what's going to happen is basically this is going to go into the it's going to go into the ER. And so it's basically what it should look like is it should look like this. If this was basically it's going to look like this at some point. I wish I drew that better, but you get the idea. But it's going to look like that. And so what's going to happen is this is going to get cleaved and this is going to flop into the cell. So which is how you end up with both the N and C terminus both in the ER lumen. Hopefully that kind of makes sense. Yeah. Not gonna harp on this too much. Already talked about this a little bit. Yeah, I already mentioned this in the previous one. Okay, this RAN, nuclear import. So RAN GTP is another Jeff combination with its stuff like that. And RAN is important because it's used in nuclear import / export. So basically, let's see if I can find a nice image of it. This is a good image of it, actually. Yeah, wait, this is a good image of it, I think. This is okay. But now we're talking about this sequence, the nuclear localization sequence. This is talking about that. So we're moving on now from the hydrophobic sequence. So now we're talking about the nuclear localization sequences. So keep that in mind. We're switching gears a little bit. So looking at this, nuclear import, RAN GTP, and nuclear import receptors. The one thing I want you to take away from this is that RAN GEF is only found in the nuclear side of the membrane. And so what that means is that RAN is only active in the nucleus, right? Because in order for a G protein to be active, it needs to have its GTP. So, and the other cool thing about RAN GEF is that RAN GEF is actually bound to chromatin. Just a kind of a cool little fact. So, again, remember, in order for there to be any transport of anything into the nucleus, it's got to have this sequence, which is positive charges. Or sorry, it doesn't have to have this sequence. It just has to be positively charged. So any mix of arginine and lysines. And so now we got to talk about the importance of how proteins are transported into the nucleus. The way that this works is you're going to have a protein. That protein is going to have a nuclear localization sequence. And once it binds that nuclear localization sequence, once that nuclear localization sequence is, there, what happens is a nuclear import receptor, which is another protein type, will bind to the protein's nuclear localization sequence. Once that import receptor has bound, you can do a whole bunch of other stuff, basically. There's also other ways of doing this. So, like, for example, here's another example. In this case, you have a protein that has to bind to an adapter first before binding to a nuclear import receptor. So there's many ways of doing this, but I'm going to focus on these for now. So we have our nuclear localization sequence, we have our import receptor, and we're now here. So this is a very interesting cycle, and remember, RAN is only active in the nucleus. It's not active anywhere else. So, how the heck does that work then, right? That's the real question about how this works. So, let's start with this. So let's say that starting from the nucleus, right, we're going to take our RAN GTP and we're going to bind it to a import receptor. Once this is bound to an import receptor, this import receptor is responsible for basically well, I guess I should probably start it from here. Let's start it from up here. I think it's actually better to start it here. Okay, so nuclear import receptor. It's blank. It has nothing in it. It's in the cytosol. Then the protein with the nuclear localization sequence binds to this receptor. This receptor and has a sequence that is recognized by the cytosolic fibrils of the nuclear pore, and that pulls it into the nucleus. Once it is in the nucleus, RAN-GTP binds to the second slot in this receptor, and it causes a conformational change, which results in the release of the protein. Once that protein is released, we then have this RAN-GTP receptor, and so we now have this thing. This then gets pulled out of the nucleus into the back into the cytosol, and when it's back in the cytosol, the RAN-GTP hydrolyzes its GTP to GDP, and that causes it to dissociate from the receptor, and you're back at this starting point. And so now you can repeat the whole process again. You can bind another protein, you can pull it into the cell, you use RAN-GTP to kick it off, and then you restart the process again. So you can keep doing it over and over again. So this is specifically for nuclear import. Now for nuclear export, let's see, where do I start with this? Let's start with the nuclear export receptors. This is a very similar process, but now we're talking about the nuclear export receptor. So this is in the, I wish it showed me. Yeah, this is in the cytosol right now. So we're in the cytosol, right? What's going to happen is we're going to go into, this is going to bind to the fibril. It's going to get pulled in. The RAN-GTP I actually want to double check something. Wait, a separate transporter. Oh, this is very important, actually. This is not that important to, like, it's not important to know how this works. You just need to know that. You're probably thinking from this, oh, well, isn't RAN just going to get stuck out here? Yes, you're right, but there is actually a separate transporter that returns RAN-GDP to the nucleus so that it can be exchanged for RAN-GTP by RAN-GEF. You don't need to know the specifics of that, though. And also this, the hydrolyzation of this GTP to GDP occurs because of RAN gap. Okay. Anyways, back to the export. So now for nuclear export, remember we used leucine, isoleucine rich sequences. So remember, for this up here, we used basic amino acids for import. For export, it's leucine, isoleucine rich. So not just hydrophobic. Hydrophobic is ER. Leucine, isoleucine is for export from the nucleus. So still uses RAN though. So we're going to start with this. Nuclear export receptor. Similar process. It's in the cytosol. Gets pulled into the nucleus. From here, RAN binds to the this nuclear export protein. Once the RAN binds, it causes a conformational change that causes a protein with the nuclear export signal to bind. And so now you have this thing that has a RAN and the cargo on it. This is then released from the nucleus into the cytosol. And what happens now is, again, the RAN hydrolyzes GTP using the gap. And once it's hydrolyzed, this leaves the cell. Or not, sorry, what did I say? Not leave the cell. Once it's hydrolyzed, the RAN dissociates, and the RAN dissociating causes another conformational change that kicks off the cargo. And so you're back to this free export protein. And we do the process again. Come inside, RAN binds, then the cargo binds, then the RAN and cargo plus the receptor leave, RAN hydrolyzes GTP, the RAN gets kicked off, and because the RAN gets kicked off, the cargo also gets kicked off. So, yeah. This is a very interesting slide. I'm going to be honest with you, I don't think this is that important. You guys will talk about translational modifications later, post-translational modifications later, but there's many different types of modifications. There's glycosylations, there's blood type. So blood typing is actually caused by glycosylations. There's phosphorylations. There's all sorts of stuff. There's cleavages. So a bunch of stuff can occur based off of
for post-translational modifications. So, yeah. I had the hiccups now. Jeez. Okay. Well, I hope this video was helpful, you guys. We're actually right on 30 minutes, so yeah. If it was helpful, great. If it wasn't, oh boy. But, feel free to leave comments, ask questions, come to the IC, do whatever. More than happy to answer questions. But other than that, yeah. Have a good one, you guys, and happy studying.